Okay, so a little earlier in this topic we talked about how water moves in and out of the cell. Remember, water always moves down its concentration gradient, with its concentration gradient. So it always moves from where there's more of it to where there's less of it. And in order for water to be able to actually get in and out of the cell, it can't just pass through the phospholipid bilayer. It has to go through these aquaporins, these membranes, proteins with kind of like a tube or a channel down through the middle of it that allows molecules like water to get in and out of the cell. Okay, and that's called facilitated diffusion. We also talked about how the facilitated diffusion of water and water alone is called osmosis. Right, so bearing in mind that we've covered this, I want to go through this in a little more detail so that you understand a couple of other things about how homeostasis of a cell water content is maintained. So how do we keep the water content steady in the cell? So we need to know a couple of terms. And the terms are hypertonic, hypotonic, and isotonic. And in this diagram, this is a very, very simple representation. So this circle here represents a cell, and this so this would be the cell membrane. And I want you to remember cell membranes are selectively permeable. That means they regulate what can pass in and out of the cell. Okay, so this is the cell membrane surrounding a cell. The blue dots represent water molecules. The red dots represent something else. It could be salt, like sodium ions or something like that. On this one, if you look at it, it is very obvious that there are more water molecules extracellular, more water molecules outside the cell, and fewer water molecules inside the cell. Okay, intracellular. So extracellular, intracellular. And I could count all these water molecules up if I wanted to, and it would show you that there's more outside than inside. Water moves down its concentration gradient. So water is going to move from where there's more of it to where there's less of it. So water is going, in this case, will flow into the cell, okay, until there are an equal number of water molecules in the cell, with the blue ones and the red ones will be the same number. Okay, because there's more water outside of the cell, right, than inside, we call this term hypotonic. Hypo means less in the cell. Okay, and water is going to flow down its concentration gradient. So if you put a cell in a hypotonic environment where there's more water outside than there is inside, water will move down its concentration gradient into the cell. Now, if the cell is filling up with water, the cell itself is going to expand. I'll come on to that in a minute. Now, let's look at the one in the middle, because this is a kind of a different situation, because it's very obvious that there's more water inside the cell than there is outside the cell. This is known, because there's more outside, this is known as a hypertonic. And hypertonic, and that refers to what's inside and outside the cell. Okay, so this is hypertonic. There's more salt solution outside, or more salt molecules outside, and less water molecules. Now remember, water always moves through the semi-permeable membrane down its concentration gradient. So this time there's more water inside and less water outside. This time the water is going to do the opposite. It's going to go from where there's more of it to where there's less of it, and the water is going to move out of the cell. Okay? Now, if the cell is losing water this time, what's it going to do? The cell's going to get smaller. It's going to contract. Now, when you look at this one here, now there is an equal amount of water inside and outside the cell. So it's already balanced. And this is called isotonic. Okay? Now, isotonic solution, the concentration inside and outside is equal. Iso means the same. And so now, this is interesting, because you can't say, okay, the water's not going to move. But here's what's going to happen. For every water molecule that moves out, there will be another water molecule moving in. So if this water molecule moves out, then another water molecule is going to move in. And what we say is there's no net movement of water molecules. Okay? So, now why is this important? Okay? If you've ever been to an emergency room and they've said you're dehydrated, you are probably in this situation. You've lost water because you're dehydrated. There's less water outside the cells, so the cells have lost water. And then you have lost that water by, exhaling and sweating and stuff like that. So they're going to say to you, okay, we are going to put, they put that thing in your arm, the line in your arm, and they then take a bag of clear looking liquid, and they hang it up and they let that drip into you to help you rehydrate. Okay, it's really, really important that they get the same concentration of that solution is inside your cells as is outside your cells. So they're going to want to make sure that they use an isotonic solution. And the material they use, it's called Ringer's solution because it's made up of all the minerals and sodium ions and potassium ions and water molecules that you naturally find in your blood plasma. And that's really important because of this reason. You remember here, well, let me show you what it would look like on a red blood cell. So red blood cells are what we call biconcave discs, which means that if you look at them as a bird's eye view, they're circular, right? But if you look at them on the side view, they're kind of like that shape, okay? So it squeezes in the middle. All right. If you put a blood cell in a hypotonic solution, remember, water's going to move into the cell. What that blood cell is going to do is it's going to get bigger and bigger, and it will look like this. And what will ultimately happen, like when you blow a balloon up and you keep blowing and you keep blowing and you keep blowing, that cell will burst. Okay? So the last thing you want is to be dehydrated and have a hypertonic, like pure water put into your blood, because that will make your cells swell up and they could burst. Not a good thing. Now, what's going to happen if you put a hypertonic solution? Let's say you put a really strong salt solution into your blood through that line that they put in your hand. Well, we know that when you put a cell in a hypertonic solution, water moves from where there's more of it to where there's less of it, down its concentration gradient. And so if the cell's losing water, this is what's going to happen. Your cell is going to shrink. And if your red blood cells shrink, right, then they're no good at carrying oxygen. You do not want to do that, do you? But if you put red blood cells in an isotonic solution, what's going to happen is they will lose some water, but they will gain some water. And so your red blood cells will stay the same. So there will be no change. So the next time you go to the emergency room and they have put a drip into your arm, they're going to use something called Ringer's solution. And Ringer's solution is an isotonic solution, and it's isotonic to your red blood cells and your blood plasma. And that's so that they don't end up either having burst cells or shrunk cells. Okay? Thank you.